Thank you, everybody, for being here. We have an announcement about the MPG retreat, which is in February. We're seeking volunteers to join the Scientific and Social Planning Committee, so please take a picture with your phone or get that QR code to sign up. Another announcement, next week was meeting-free week, so we don't have primer, but the week after that, we will have a primer from Pamela Robertson from the Broad Clinical Labs, talking about clinical genomic resources. But today, we have two speakers from the Iqbal Lab, and I'm sorry, Seulki Kwon is a scientist, one, not a postdoctoral associate, so sorry about that. Let's see. I had these. Okay, yes. Okay, so Seulki is a research scientist in computational biology with a focus on protein bioinformatics and variant interpretation. Her research focuses on understanding how genetic variation alters protein structure and function using computational models to uncover mechanisms of disease. And then we have Dr. Yoochan Myung, who is a postdoc. His research focuses on developing pipelines to interpret structure-function relationships and base editor tiling screen data. He integrates AI, biophysics, and structural biology to interpret variant effects and guide protein engineering. So great, we'll turn it over to you guys.
Yeah, good morning, everyone. It's a pleasure to present our Genomics to Proteins portal in the MPG session. GTP Proteins portal is a discovery portal designed to help researchers to link the genetic screening outputs to protein sequences and structures. Our team is in the Bioinformatics and Machine Learning group under the Letters to Cure Accelerator at the Broad Institute. Before I dive into the portal, I'd like to acknowledge that this was a truly team effort, and I'd like to thank many our collaborators and funders who made this entire project possible and valuable, and thanks to our scientific illustrators as well to create a beautiful visuals for the portal. And this work is published in 2024. So if anyone is interested in more, please please feel free to check out the papers as well. Genomics to Proteins portal started on fundamental questions that how we can connect genetic variations all the way to the protein structures. So let's say a geneticist identified a genomic variant on the genomic coordinate. It points out the mRNA sequence change in a specific transcript, and then it translates into the amino acid change and ultimately impact the protein structures on the 3D space. So understanding this entire flow of the information is crucial to understand this to understand the disease mechanism and thereby build biological hypotheses and therapeutic strategies. So understanding how these protein coding variants affect protein function is crucial for translating genetic genomics discovery into the clinical insights as well. And here's an example of the DXCX gene, which encodes RNA helicase. And researchers have identified 5 de novo variants from the patient, 5 de novo missense variants from patients. And when they mapped the variants onto the protein 3D structure, you can see that they are clustered on a certain regions of the protein, which turns out to be really important functional motif of the protein, which is the RNA binding motif. So this immediately suggests that this mapping reveals the mechanisms and suggests the mechanism that this disrupting this RNA binding ability would drive the pathology of the disease. And this is not only case study, we have many more genes where we see a disease mutations cluster together and impact specific protein functions. Uromodulin kidney disease mutations are clustered in the N-terminal part of the protein, and MORC2 mutation is on the protein-protein binding interfaces. And more interestingly, in case of the GABA receptor, you can see the mutations clustered in different regions of the protein actually display a different severity of the phenotypes as well. So when you actually start looking at the data and the data landscape these days, you'll notice there's an incredible amount of data available. First, genetic variation. Thanks to the high-throughput sequencing technologies, now we can identify clinically relevant variants, and they are all deposited in the ClinVar databases, and we have a huge number of human population data, population variation deposited on the GNOMED databases. And interestingly, if you look at the clinical significance identified as a function of time, you can see the number of variants of uncertain significance is growing really fast compared to other variants of turns out other variants of pathogenic or benign. So that highlights the need for the better variant interpretation tools. Second, structural biology. We now have over 200 million experimentally resolved structure in the protein data bank, and AlphaFold is creating millions of proteins data. So that suggests we now have an unprecedented coverage of the protein structure for the human proteome. And lastly, functional genomics, such as deep mutational scanning experiment, is now testing like hundreds of thousands of variants at the same time, creating a rich set of functional data as well. So each of this field is extremely valuable on its own, but the real power can come from when we actually integrate this data. So our central question was how we can usefully integrate this data, and this is what exactly the Genomics to Proteins portal is aiming for. So this our Genomics Proteins portal combined multiple databases and provide this holistic identifier mapping. So if you query a gene, you can see the connections of this ensemble transcript and RefSeq to the UniProt protein isoform identifiers and to the structures as well. So if you, and then we map a variant on those transcripts seamlessly all the way to the protein's 3D structures, along with other protein features that we integrated as well. So currently, the portal integrates the genetic variation from ClinVar and HGMD, which are disease-related ones, and also human population variants from GNOMED. So altogether, we're currently aggregating more than 49 million genetic variations for the entire human proteome, not only including single nucleotide variants like missense, nonsense, synonymous. We also have non-SNV mutations as well. And we also provide the database-specific filters to make this interpretation intuitive. So you can actually see variants by their molecular phenotypes, and plus you can see the GNOMED variant by allele frequency or count. And for clinical ClinVar variant, you can filter them by the clinical significance. So you can see, like, oh, I'm interested in pathogenic variant, I want to see the benign variant. So you can like opt in and out from the portal interface. One of the key strengths of the portal is that we have like the highest structural coverage. So the AlphaFold protein structure prediction covers the structure of more than 12,000 proteins where there's no experimental structures available at all. So if you look at the chart, we are able to map more than 70% of the GNOMED variant, 50%, 40% of the ClinVar and HGMD variant where there was no structures available. Then we can retrieve them by incorporating the AlphaFold databases. So altogether, in total, we were able to map over 90% of the variant on the entire human canonical isoforms successfully on the protein structures. So now we have a genetic variants covered, and what next step is to layer them on the protein feature databases. So we collect many different protein features from different databases, and we provide them as a category of 6 different protein features. So I'll just briefly introduce what that means. So physical chemical properties about like the side chain chemistry of the amino acid, like such as polarity, hydrophobicity. And we also have a full set of pre-computed structural features from AlphaFold and PDB. So you can see the secondary structure annotations, solvent accessibility, and importantly, the confidence of AlphaFold score, and also the pre-computed potential binding sites on the protein surfaces as well from the egg pockets. And also from the structure, we pre-compute the protein interactions within the protein or between the protein, which is these 4 different types of chemical bond that you can identify within the structure. And we also bringing in the rich set of UniProt sequence annotations, and they mainly focus on like the active site, binding site with important functional sites and domain annotations, regions, motif. So you can pull these UniProt annotations on the sequence viewer all together with other features as well. And we bringing in the post-translational modifications from the Phosphocyte Plus databases as well. And lastly, we work with the MaveDB, which is a public repository for data set from this multiplexed assay of the variant effects. So if the protein has the Mave data published in the literature, they catalog them, and from the G2P portal, we visualize them as a heat map, and it's currently available in about 500 proteins. So we have 2 different modules from the Genomics to Proteins portal. First one is a resource module where you can search your gene or protein by their gene symbol or UniProt ID, so that you can access the entire resources that we put together in the portal. And the second module is the interactive module, where you can actually upload your data, your mutation, your annotation, and work with the known protein structures, or even you can upload your structure to interactively work with the data. So I'll briefly, I'll just walk you through to the resource module, to the portal. So in the research module, as I described, you can search by gene. So I'll just search the gene, and it'll take you to the landing page. So from the landing page, you can see the generic information about the gene and protein. So it has a cross-reference to external links to GeneCard, HGNC, and Panther. And if there is a known gene disease relationship established by GenCC, you can see that, and you can visit them as well. And the UniProt link is provided, and it provides the canonical protein isoform identifier. So if you go down, we have 5 tabs. The first tab is for our sequence viewer. So this is one of the main features of the portal. So then you can bring in and filter in and out many different protein features on the track. So you can click whatever protein feature that you are interested in, and you can always zoom in and out viewer so that you can click your residue of interest. And this is by default provided for the canonical protein isoform. But if your protein of interest is non-canonical protein isoform, such as this -2 protein, you can click it, and then we align it then for you on the sequence viewer, and you can see there's a gap of this in the non-canonical protein isoform, but you can also translate this full set of protein feature annotated here to the non-canonical isoforms as well. And you can always screenshot click by clicking this to download these high resolution images of the sequence viewer that you customized. And you can also download the annotation as a CSV file so that you can bring in a table and analyze from your own pipeline. And also you can just see the tables here on the website. So click the view table, you can just see like full set of features aligned by the protein residue ID. And I'll visit this fourth tab, which shows the table of our identifier mapping from gene to transcript to the protein isoform. So you can see what is the canonical isoform for this gene, annotated with asterisk. And you can see what's the canonical ensemble transcript here. And you can see also for the RefC, you can see what's the main select transcript, where many of the clinical variants are annotated on. And go to the additional resource. You can see the external link to other databases about this gene, such as UCSC, Campbell, Drug Bank, and the disease annotation database like OMIM and Morphonet. And when there is a MAVE data available, so I'm bringing in a case study gene where the MAVE data is available. If you click the additional resource tab, you can see the preview of this MAVE data as a heat map, and you can always select the data sets relevant. And it also directs you to this reference databases of the MAVE, so then you can look at more detailed data and download it. And let's go to this, let's see the variants now on this sequence viewer here. So I'll go to another case study genes, which is a tyrosine kinase 2. So if you click this variant to protein sequence tab, you can still see this same sequence viewer that you have looked at, this protein sequence annotation tab. But in this tab, you can bring in the variant data. So you need to select the transcript here for the mapping. And then now you can see there's a nomad variants categorized by their molecular consequences and allele counts and allele frequencies. So you can click. I'll just show you the multitude nomad missense variant here. And sometimes your transcript where all of these clinical variants are annotated is not always encodes the canonical protein isoform. So in this case, this -2 protein is corresponding to the main select transcript, and now all of this clinical data is annotated on this transcript. So in that case, to address this, we provide the mapping and aligning variant from another transcript. So if you click this button, you can select another transcript to be aligned. So I'll click here to the main select transcript. And then now you can see this ClinVar variant annotated on another protein, which has a gap in the middle, now aligned to the canonical protein isoform. So you can see those variants now with the full set of protein feature annotation annotated on another protein sequences. And you can also view table here to see nomad variants and has informations, and also the ClinVar variants as well. Yeah, there's no record because we selected the canonical protein isoform. And now you can map those onto the protein structure. So if you go to the third tab, variant to protein structure, we list the list of the PDB entries and AlphaFold entry. So you can always sort them by the method and resolution or residue count. So if you are not sure which structure to pick, I suggest you to pick the most covered PDB structures. But we also have an AlphaFold structure which provide the full length structure of the protein as well. So I'll go with the AlphaFold structure, so you can click any rows that you want. And we are now seeing the AlphaFold structures here in the structure viewer along with the sequence viewer. And you can see another structures of this protein. And if you click it, you see how these are aligned on the protein sequence space. So for example, this 7L0 PDB is only covering those regions of the protein out of this entire length. And I'll just, drop it in. And if you click the nomad variant, I'll just click the missense nomad variants, not aligned one. And I'll show you the multitude. and maybe all of them is very rare nomad variation. And now we have an aligned missense variant from the -2 protein. And I'll click also the missense variant and only a pathogenic clean bar variant. So if you click this track now, you can map them onto the protein structures. You can map it all together, the clean bar variant together with the nomad as well. And you can also bring in other protein feature annotations, like if you click this amino acid property, you color by the property, surface area, alpha fold confidence, and the PPIs and active sites. So I'll show you this variance along with the domain annotation. So from the UniProt, it's annotated at the protein kinase domain here. If you click it, you can see there's a light blue area where this domain annotation is located. And if you can make a full screen, you can see that this nomad multidome variant is kind of spread all over the protein structure, but this clean bar pathogenic mutation is mostly populated in this protein kinase domain. Not surprisingly, it's important functional domain of the protein. So if you disrupt this region, you'll get a disease. Yeah, and you can download this entire visuals that you customize through the PyMol script so that you can work further for your publication quality figures or other analysis available in the PyMols as well. Yeah. So this was a research module. And we have introduced another module, which is the interactive module. And I'll hand it over to Yoochan to present and walk through the interactive module.
So from here, I'm going to introduce the interactive module of Genomics to Protein, or shortly G2P portal. But I'm going to want to start with asking questions. How many people here have experience of mapping your experimental data on 3D structures? No? Right, so I didn't expect that. But if you have any experience, it's not very easy. I mean, if you have a decent script, it's quite easy to just use it to map your screen data or experimental value onto the 3D structure. But in short, it's quite tough. It could be very error-prone process. I'm going to highlight two things on this interactive module. The first thing is this interactive module of Genomics to Protein portal is super easy to use. Literally, you can just copy from Excel spreadsheet and paste it, and then we can visualize it on the protein structure. The second part is, I guess this just browsing the available research on this protein is important, but more important application is mapping your data and then actually see alongside with the other features that the Seulki introduced. So you can actually analyze and interpret better in the variant or other structure feature, so that can help you to understand your experimental data. So I'm gonna walk you through just one by one. So the interactive module, you can click this on the right-hand side button, and then you see the two sub-modules here. The first case, the first sub-module is when you have a gene or protein identifier, like a UniProt ID, and you have your own experimental value, like an Excel spreadsheet, then we can start with the first module. So I'm gonna actually have a example DNMT3A, which is DNA methyltransferase gene. So if you type it, it'll show you the canonical UniProt ID here. So which looks correct, so I'm gonna proceed it. Then it will show you all the available protein data bank DNMT3A structures, but only for the human, not human. Non-humans are not here. Non-human proteins are here. So I'm gonna select those one specific structure, because the later part, I'm gonna introduce the data, which is mutational screening or functional screening data. And then I want to see that the mutational data with a homodimer of this DNMT3 protein. So I'm going to select that PDB has homodimer formation. So if I click this PDB, you can actually see, make it bigger. Yeah, it looks homodimer here. And you can also check the coverage of this DNMT3A gene and their corresponding PDB here. So it actually covers half of the DNMT3A protein. And you can see that two chains are here. And you can see the color corresponding chain A and chain C. But if the coverage is not enough, I mean, if you're more interested to see the full coverage from AlphaFold structure, then you can just move on to the AlphaFold tab, and you can select the AlphaFold structure to map your data. Right? So I'm gonna just keep with the homodimer. Then we're gonna have this annotation viewer. So here, what we're gonna do is putting the experimental data. So, okay. Let me open the Excel. So this is a data for this mutational function scanning data. I'm gonna just copy whole spreadsheet data, and then I'm gonna paste into here. And then submit here. But if you're not very familiar with this, the form or formatting things, you can actually click here, see the example, how the formatting should be followed by this kind of format we define here. So it could be just comma separated or tab separated, but you can have any kind of format, any kind of headers like this. I'm going to show you more real case scenarios. So if you submit your data, then the portal automatically process it, and then it'll show you this kind of screen. Then in this step, what you need to do is check whether this data is correct or not. So for example, the first column has to be always the residue index. It's because Genomics, the protein portal needs to need to map your data with this column information to the other Genomics to protein research data. So this column has to be always residue index. But you can actually have different column, not just position, but something else. Right. And then I'm going to explain three things. So you can see that buttons have a different labels here, mutations and scores and features. Each data type has a different representation. So for example, if I set this human residue column with this mutation data type, which requires specific formatting. So I'm going to just quickly show you what formatting this mutation column requires is this first row. So it was actually invented for showing information with sphere representation in 3D viewer. We're going to see in the next page. Here, I actually follow the last formatting. So any letter, single letter, here. So you can see that the second column has one letter, which is wild type residue letter. And the third column is score. If you put any values in this score column and then define as a score, then you can color your protein with the gradient or continuous gradient color. So like a red to blue, white to blue, anything. And the last type is the features. So feature column accepts either value or string or text. So I'm going to show you one of the examples here. I'm just going to have it here. So I already marked some residue of interest in their location. Oh, these residues are cofactor binding site. So I actually typed in there already. And this is a residue in homodimer interface. the last column is another way we can show something. So because the feature column will color this protein structure, but if I have it as a mutation, then I can actually change the representation with the sphere. So you can just play with your columns with a different data type to show in different representation in 3D structure here. And importantly, if you have any typos or something, you can actually modify from here. You can change it, except except this headers here. Okay. So once you submit your annotations, then you can see that the structure we selected here, and this is colored by based on the selection of events, the left sequence view. I'm gonna color this one based on the score of the score screening the data that I feed in. You can see that the darker purple color is representing higher value of the screening data. And because there are so many color or screening data here, and then I actually marked very high scoring region with mutation data type notation. So you can actually see that with the sphere as well. But those are having spheres have very darker purple on the protein cartoon as well. So with this information, we might be able to interpret some important biological things, but I'll further go with adding the genomics to protein research features. So if you click this view additional annotation, that is way we can add features from research module. So first, I'm gonna map clean variance information. You can see there's no information available because clean variance informations are mapped onto the second transcript here. So if you click the second transcript, then now you'll see that clean map on the second. So I'm gonna select missense, but only pathogenic variants. So now we can see that 60 pathogenic variants on top of this, the scores of our experimental data. Some of them are aligned well, some of them not. So it actually gives us like some kind of information why how the mutations align with the pathogenic ClinVar mutations. But I'm gonna actually add a few more features, such as potential small molecule binding site, which is from F-pocket prediction. You can color the protein structure with this F-pocket, and you can see that those greenish part is where the small molecule can bind. And another important thing could be here, because I said like I'm interested in to see the homodimer context. You can see that there are 3 features available which represent the protein-protein interaction site, which is coming from analyzing all the DNMT3 PDB and finding protein-protein interaction residues. Okay, I colored them here. So this mustard color is showing protein-protein interface residues or interacting residues in DNMT3 protein. And so I want to actually see that whether these high scoring residues are aligned, then you can see that, oh, okay, these 2 residues could be very important mutations in terms of DNMT3 function, because this is actually happening on where the protein-protein interaction happening. Right. Okay, so, all right, and those are the features we can edit on, and then you can also change this color by using this edit colors and select corresponding features. You can change the scores, a color, not blue, purple, but different colors here. Okay. And as Seulki mentioned,
can I ask a question? Sorry to interrupt. I'm wondering about what to do when you're mapping your mutations on your favorite protein of interest, but your mutations are not like obviously segregating to a certain area. I know that's kind of an open-ended question, but what kind of statistical tests could you run? Like taking into account the 3D structure. I mean, some of the things that you showed, like the TLK2, it's like nice when it obviously all goes to the kinase, but usually they don't. So how do you approach those scenarios?
So you're asking like how we map the mutations of the protein?
No. How do you interpret
Interpret
the mutations on the structure when what you think are clinically pathogenic variants aren't mapping obviously to a domain?
Yeah, we actually have many case studies where we don't have a clear clusters, of course. So I was going to preview a little bit, but we are actually designing a scoring system for a variant where we bring in all these protein features and see what are those features are actually relevant to the pathogenicity, so that we can provide or help to build a hypothesis. So, okay, this variant is not clustered, but it has this features, and it has a certain area in the protein 3D structure, so that it may have an impact on protein functional side, or it can be from, okay, this is a disrupting physical chemical properties. So we are actively working on this variant interpretation tools, and is about to incorporate it into the portal. But if you have any interest of your gene, you will always you can feel free to reach out to the group so that you can collaborate. We always welcome collaborations. Yeah.
Thank you. But yeah, we don't actually give any scores here but we can actually compare with those pathogenic variants on top of your mutations. Okay, we can actually export these annotations, and then we can actually analyze in the text or table formats. Here, I want to actually show you another thing. So the cool thing from here is we can actually go back to the structure, and then we can actually interpret this one in the different contexts as well. Let's say DNMT3A actually methylate the DNA, right? And then we can actually check whether these mutations are close to the DNA as well. So what I want to try to show you here is, even if you go back to the earlier steps and then click a new PDB structure, it actually kept all the information what I did. And then actually you can actually keep all the annotation as it is, but you can change the structure. So you can actually have another interpretation by changing your structure in this interactive module. The last part here I want to show you is a sharing function. So if you want to keep this session active for next 7 days, you have to click this share button, and then you have to put your Gmail account and edit. Also, you can actually share this one with your collaborator by putting their emails and create an account, create a link, and then you can just copy this link and share it. So if I paste in here, then I can keep this session for next 7 days. Okay, I think that's pretty much everything from here. Do you have any questions so far?
Any questions? There's 2 questions. Well, one is, how do you deal with, or can you deal with in-frame deletions? So, or exon skipping. I work on a large autism cohort, and we're finding a lot of these types of variants.
Yeah, we in the sequence viewer tab, you'll be able to access the in-frame deletions and frame shift variants. But you can map those positions on the protein structure, but interpretation is that since that position, your protein sequence is going to be entirely changed. So you might have to aware of that this interpretation onto the protein 3D structure might not be that accurate, but we can pinpoint where is the mutation, but other functional annotations and this PPIs and structural feature might not be biologically relevant. It should be really case by case. You need to look at where the sequences are going to change by your non-SNV sequence mutation.
Gotcha. Yeah. Another question on the channel here is, someone's asking about publication-ready images, if you could just kind of show that one more time, because I think that's of interest.
Um, oh, yeah. So yeah, we can export this one here as well. There are a ton of ways to export the images here. I also personally export this session. If you're very familiar with this Moorestar viewer. So this is
What's that?
This is called a Moorestar viewer, which RCSB data bank also use it. So if you actually save this session, export it, you can keep this exact same view for the later.
Can I add to the
Yeah,
answer to
please,
that
please.
question? So if you go back, Yoochan, so folks can also download the PyMol script. So all the mapping that is performed, they can download and open locally in PyMol session. And that is the best way to generate a publication-ready figure. But from the portal will be this structure viewer. And then you can look at the screenshot over there on the screen viewer. That takes the screenshot of the entire sequence viewer.
Gotcha.
So the sequence viewer and structure viewer side by side. And if you want to deep dive, then what I recommend is to download the PyMol session, open it in the PyMol and then.
Cool. All right, great. Thanks.
And that PyMol section will have all these features that side tab. So you
Okay.
can actually have almost same features there.
Very good. Cool. Question?
So right now you have showed about single protein and how mapping, variants to a single protein. Is there any plan to get into protein-protein interaction and having interface between these two proteins and mapping those variants to it?
I actually put that.
Yeah, you can bring in any PDB structure if you have a complex protein structure, chain A and chain B, and chain A is your interest of protein, and chain B is another one. If you go, can you pull up it? PDB structure with other chains?
Such as, yeah, 3A.
Yeah.
Sure.
Yeah, if you load this heterodimer complex structure with DNAs and everything, you can see actually the chain information here. So this chain K and P is the MT3A, and chain N and S is the other protein. So you can map protein and see where those mutations are located. And from these PPI features, this is the pre-computed ones that we calculate from all available PDB structures. So if you click it here, like interchain hydrogen bonds, and you zoom out, and you can see the hover-over text here. So it says to annotation. I'm not sure if it's working. You can see the hover-over text to annotation, which says K299 and K343 is interacting, and F303. So even if it's between chains, we annotate them in this tab so that you can download it actually later for your analysis.
Great.
This is an amazing resource. So I saw that you have already mapped all the nomad and clean bar variants into this, all the protein residual in this portal, but is it possible to map, say, any arbitrary genomic variants, for example, that's found in a new data set and map up into the protein structure here?
Yeah, that's exactly what this interaction mapping module does. So he uploaded this base editing screening data, but as long as you have a right format, which is HVSP like formats or the single amino acid letters, you'll be able to map any mutations. So if you go to here, just click any case studies, any structures. In this annotation tab, you can just arbitrarily say, okay, this is residue ID column, and this is your mutation, and like, I don't know, position 34, Y34K, something like this. That will just, take you to this mapping from the simple.
Too much about like this age.
You're talking about genomic coordinate.
Yeah, is it possible to become say the GRCH38 and first do this mutation? What are the recommended?
In that case, I recommend to use the VAF ensemble VAF tool so that you can upload your genomic coordinate and it will give you the protein consequences. And we only support the protein consequences at this point. Yeah.
So yeah, this interactive module supports just protein level mapping. Yeah.
There's a question on Zoom. Do the annotations you map to the structure include post-translational modifications, including recognition of glycosol transferases, ubiquitinases, et cetera?
Yeah, we are bringing post-translational modification from the Phosphosite Plus database. They collected, experimentally validated, and predicted the Phosphosite Plus size. So as long as the data's there in the database, you can bring it to the portal and see in the 3D space.
Cool.
Yeah. I think we are running out of time. So I hand it over to Seulki for the finishing of the
Yeah. So
Great.
we have another module which you can actually feed your own protein structures as well. We are skipping this, but feel free to watch this tutorial video that we have. We have a full playlist of YouTube tutorial that you can follow about various functionality of the proteins of the portal. So go back to the slide. So I know many of you work with the confidential data and worry about like uploading your data to anywhere else. To address this, we care about the security and confidentiality. So we built our infrastructure so that your user uploaded data never leaves the user's local device and circle on the front end. So it means it never shared or stored through the backend server of the portal. And your interactive mapping module requires you to the Google sign-in authentication. So it adds another layer of security that only verified user can upload and access the interactive mapping module. So for developers and anyone interested in incorporating this genomics to proteins portal to their own pipeline, we provide the Swagger API interface where you carry your gene and then get some of the features from the portal. And we have also Python client library. So you can see this API tab for more information. And another thing I want to mention is that we maintain and update the portal in a regular basis. So some of data, which is really time sensitive, we dynamically load them, such as the list of PDB available. So as soon as it's deposited in the PDB or AlphaFold, you'll be able to access the structure. And some of the pre-computed data, like structural data or the PPI data, we have a major biannual update. And for the CleanVar variant, given that it's rapid speed of the data submission, we update the CleanVar variants on a monthly basis. So if you go to the release log tab or to the portal, you can see all of this version information at a particular point and all of this log that we updated so far. Today we have more than 10,000 users and researchers around the world, and this platform continues to grow. We continue to expand our functionality to different directions, and I just want to highlight a couple of directions that we are actively working on. First is the question that we are developing the variant scoring system from the protein's 3D structure to interpret this variant by their protein level features and like which feature actually attribute that. I'll have another slide for this to briefly introduce it. And we are also thinking, working on this agentic AI system that you can query from this large language models, natural language basis. And we have also working on this integration of the on-demand tools so that you can actually run a couple of tools on the portal and analyze your data as well. So just quickly introduce this scoring system. We had a question like, so which protein features are actually enriched within the pathogenic variant affect the pathogenicity? So there's no time to explain it in detail, but the main idea is that we curated a data set of the pathogenic and proxy benign, and with the feature enrichment analysis, and created these types of in silico scoring systems so that you can query a mutations. For example, I searched the PPAR gamma R194 YW mutation. It will give you a score, and it'll give you the interpretation based on the score. This is likely to be pathogenic with a certain confidence. And by the nature of the design of the score, we can actually decompose the score into different directions of the protein features. So you can actually measure where is the score coming from, is coming from biochemical property change, or it's coming from the structural stability aspect or functional side domains. So we hope this score to provide and help to build a hypothesis. And it should be really gene by gene approach, where you have an expert knowledge and you have a score, then you will be able to create a hypothesis. So this is all about platform. You can visit our website and go to the About page to read all the documents here. And please follow our the Twitters, and you can directly contact us through the email. And as I briefly mentioned, we have a YouTube tutorials ready, so you can always visit them. And we always welcome users' feedback. So please use the feedback form here from the portal. And if you have a case study, we very much welcome to collaborate and provide a hands-on workshop that we actually did multiple times for those groups as well. And more tutorial will be coming, so please stay tuned on our YouTube. And thank you all for listening, and I'm happy to take any questions.
Okay. Any other questions from the crowd? Okay. Thanks so much for your talk. We really appreciate it. And then the seminars next door. Thank you.